Assalamualaikum and hi, my name is Nur Iman Binti Mohd Rasid and my supervisor is Dr. Fazrina Nadia. My final year project title is In Vitro Screening of Crude Extracts of Microalgae for Anticancer Activities. Cancer is a genetic disorder and caused by certain gene modifications that regulate the way our cells function, especially how they develop and divide. In all types of cancer, some of the body cells begin to divide without stopping and spread into surrounding tissues. Cancer awareness is widely spread worldwide because it is one of the major causes of death. As you can see in the figure, Asia tops the chart for mortality caused by cancer, and there are increasing number of research has been done to find solutions to this problem, especially in searching new natural-based bioactive molecules. There are a lot of contributing factors to cancer, and exposure to specific chemicals with carcinogenic effects and mechanism is one of the reasons. Recent research shows that carcinogen found related to environmental and occupational with specific exposure may increase risk for certain type of cancer. For example, exposure to radio frequency with non-ionizing radiation emitted from mobile phones can cause brain cancer. Microalgae is widely used for its nutritional and pharmaceutical components to animals and humans because of its characteristics, which are easily cultured. And it is known in various fields such as health, food, and renewable energy source. In health field, microalgae are widely considered for its ability to become a safe natural source to inhibit cancer, and it is an alternative for a more sustainable source for anti-cancer activities. There are several sources of microalgae that we can obtain, for example, from the river, pond, and lake. A need for cancer treatment alternatives as current treatment comes with greater side effects, for example, anemia and memory problems that are caused by chemotherapy treatment, increases the interest in further research of potential bioactive compounds that are able to treat diseases, and for the same purpose, microalgae is one of the natural sources that is widely focused on for its beneficial components and simpler growth requirements. Based on a number of studies done, there are a few biological activities by microalgae that has gathered attention, including antioxidant, anti-cancer, and antiviral. In this study, in vitro screening of crude extracts was investigated on how its component will benefit future society in health field. The objective of this research is to investigate the in vitro anti-cancer effects of local microalgae and to identify the bioactive compounds of microalgae for anti-cancer activities. Death and incidents that happen due to cancer are increasing worldwide, with lung cancer tops the chart, and it has also become the main reason of cancer death among men. On the other hand, breast cancer is the most frequent type of cancer diagnosed among females and the leading type of cancer that causes death. However, these diagnosed cancer and their frequency vary between countries and are dependent on one's life and social style and her or his exposure to carcinogen. As you can see in the figure, the highest incidence of cancer are lung cancer, breast cancer, colorectal cancer, prostate cancer, and stomach cancer. And the same type of cancer is known for its mortality rate. Microalgae have simple requirements for growth, and for that, they are able to feasibly generate lipids, carbohydrates, and proteins at a substantial scale and become good alternatives that is environmental friendly to the consumers. Some of the commercial uses of microalgae are cosmetic industry, pharmaceutical industry, food industry, and biofuel industry. The ability of microalgae to produce a complex molecule structure, which are hard to be achieved by chemical synthesis, makes them an attractive bioactive molecule source which are natural. A study showed important anti-cancer effects such as proliferation inhibition, invasion, and migration, as well as apoptosis induction of the ethanolic extract of an Antarctic freshwater microalgae. From the result of this research, Ethanolic extract of these microalgae are proven and expected to be a good therapeutic agent that can contribute to the development of drug and cancer treatment. Initially, the following are the methodology for my research, but I only managed to culture algae of Desmodesmus species and obtain its growth curve before lockdown. The overall methodology that should be involved are algae material and culture conditions, culturing of cell lines, preparation of microalgae extracts, treatment groups, empty cell viability assay, identification of biochemical compounds, and statistical analysis. The chart shows three different samples of Desmodesmus species cultured in AF6 medium. The light was provided and compressed air supported aeration from the lower end of the growth column. By referring to the graph, sample 1 shows a more rapid growth compared to sample 2 and 3 and are also observed in longer time frame, which is until day 10. This is because sample 1 was used as culture stock for sample 2 and 3, which was first diluted to get lower reading for day 0 for both sample 2 and 3. For next part of my research, I continue with critical review, and the following are parts of the findings from different journals that are related to microalgae and anti-cancer activity. There are several bioactive compounds from microalgae that are found to show anti-cancer activities. For example, violazanthine, fucoxanthin, and polysaccharides. The method to obtain these compounds were mostly done using a low-resolution method such as liquid-liquid partitioning and solid phase extraction, and the usage of HPLC and gas chromatography for bioactive compound determination are uncommonly found. Next, for anti-cancer activity, an evaluation of cytotoxicity for different types of microalgae against cancer cell lines were done based on IC50 value and the simplified in the table as shown in the slide. A lower amount of IC50 value indicates that lower concentration of the extract required to achieve the desired effect, and from the table we can see that granular septosis species and Syndesmus obliquus shows significantly lower amount of IC50 value compared to other types of microalgae. Based on the graph, each of the cancer cells were treated with granuloseptosis species extracts for 48 hours and then followed by 24 hours of recovery by incubating them in fresh media. Between 0 to 12 hours of treatment, it can be seen that prostate cancer cells shows the highest decrease in cell viability, while breast cancer cells shows the greatest decrease between 12 to 24 hours of treatment, and for melanoma and colon cancer, between 34 to 36 hours of treatment. However, at the end of 48 hours of treatment, it can be seen that prostate, breast, colorectal, and skin melanoma cell lines show viability below 50%, while for normal cell line, VERO shows viability decreased by 85%. Next is treatment of diethyl ether extract of Syndesmus obliquus on human liver cancer cell line, HCPG2, human colorectal carcinoma cell line, HCT116, and breast cancer cell line, MCF7. The result shows that both HCPG2 and HCT116 cell lines require lower concentration of the extract for 50% of inhibition at 42.77 mg per milliliter and 24.62 mg per milliliter respectively. While for MCF7 cell lines, the 50% of inhibition was higher at 93.77 mg per milliliter as shown in the slide. As a conclusion, microalgae is a very convenient and practical alternative that can be used in medicinal industry. There were numbers of research has been done before on the anti-cancer activity and some of the species of microalgae that showed noticeable anti-cancer effect in this review includes Granulocystopsis species and Cylindesmus obliquus. This anti-cancer effect was mostly assessed using MTT assay to observe the cancer cells' viability when treated with the microalgae extracts. And in addition to that, the biochemical compound of microalgae was also studied in this review for anti-cancer activity, and some of the known compounds are violazanthine, fucoxanthin, and polysaccharides. And in overall, this shows the potential of microalgae to be one of the future sources of medicine in treating cancer. That's all from me. Thank you for listening.